Look at your hand. Inside every single cell, there is a universe of information, a blueprint. This blueprint is called DNA, deoxyribonucleic acid. It is the code that writes the story of you, of every living thing on our planet, the smallest bacterium, the largest blue whale. For billions of years, nature has used this incredible molecule to store the instructions for life itself. It's an ancient, elegant, and astonishingly dense library. Now, imagine using that same system, not just to store life, but to store everything. Every book ever written, every song ever composed, every piece of human knowledge. This is not science fiction. This is the breathtaking frontier of biomedical engineering. The sheer scale is almost impossible to comprehend. All the data the world generates, our films, our scientific discoveries, our endless stream of digital photographs, could fit in a space no larger than a shoebox. A single gram of DNA, tiny and nearly invisible, could hold more information than trillions of compact disks. Nature's own hard drive, perfected over eons. We can finally learn to speak its language, to write our stories into its structure. The idea that DNA is a form of code is not new. When Watson and Crick unveiled the double helix structure in 1953, the world was stunned. It wasn't just a chemical, it was a structure that carried information. The 4 bases. Adenine, cytosine, guanine, thymine acted like letters in a 4-letter alphabet. This was a revelation. It showed us that life itself is built upon a digital code, one that predates our binary ones and zeros by billions of years. This fundamental insight laid the groundwork for everything that was to come. The first true leap from theory to practice came much later. In the 1980s, artist Joe Davis, with Harvard researchers, began a pioneering project. He wanted to embed a Germanic rune representing life and the female Earth into bacterial DNA. It was a poetic fusion of art and science. He translated the image into binary, then converted that binary into DNA bases. It was visionary, seen by many as art more than practical science, but it was a proof of concept. It showed a human-made message could be written into the language of life. After these early experiments, scientists took the idea more seriously. The real breakthrough came in 2012 at Harvard's Weiss Institute. George Church's team converted an entire book, text, images, formatting, into DNA. The retrieval was near perfect. So how do biomedical engineers actually write our digital data into a DNA molecule? The process is fascinating, blending computer science with chemistry. First, we must translate our familiar digital language into the language of DNA. A computer file is made of binary code, a long string of ones and zeros. Engineers have developed algorithms that convert these ones and zeros into sequences of the 4 DNA bases. A CGT acted like letters in a 4-letter alphabet. For example, 00 might become an A, 01 a C, 10 a G, 11 a T. In this way, a digital photo of a cat becomes a long specific sequence of chemical letters, a unique recipe for a strand of DNA. Once we have this DNA sequence on a computer, we need to create it physically. This is the writing process, and it's done through a technique called DNA synthesis. Think of it like a highly advanced microscopic 3-dimensional printer for molecules. A machine, called a DNA synthesizer, takes chemical building blocks of A, C, G, and T, a CGT, and carefully joins them together, one by one, in the exact order specified by our code. This process creates millions of tiny, custom-built DNA strands, each one carrying a piece of the original digital file. These synthetic DNA molecules are then collected and can be stored as a tiny white powder at the bottom of a test tube. Your cat photo is now a physical, chemical object. We must also be able to read it back. This is where DNA sequencing comes in. And just like that, the image of the cat reappears on the screen, retrieved from the molecular archive. When we compare DNA storage to the methods we use today, the differences are truly staggering. Our current world runs on silicon-based storage, hard disk drives, solid-state drives, magnetic tapes. These technologies are remarkable achievements of engineering. They allow us to access vast amounts of data almost instantly. However, they have significant limitations. A hard drive relies on moving mechanical parts and magnetic platters. It's bulky and consumes a lot of energy to run and to keep cool in massive data centers. It has a lifespan of perhaps 5 to 10 years before it risks failure. Silicon chips are wonderful, but fundamentally fragile and energy-hungry. DNA, on the other hand, presents a completely different paradigm. The most striking advantage is its incredible density. All the data on the internet could fit in a space smaller than a shoebox if encoded in DNA. Imagine replacing a sprawling city-sized data center with a single room. Our digital formats become obsolete with alarming speed. Floppy disk cassette tape. Finding a machine to read them can be a challenge. Hard drives degrade. Data can be corrupted by magnetic fields or physical shock. DNA is the opposite, inherently stable when kept cool, dry, and dark. Nature has proven DNA's durability over geological timescales. We can read DNA from creatures that lived thousands of years ago. Using DNA adopts a storage format that will not become obsolete. The key to reading it is life itself. Of course, silicon still has one major advantage: speed. Think of DNA as the ultimate archival medium. The journey of DNA storage is just beginning, and the path ahead is filled with challenges and exhilarating breakthroughs for the next generation of engineers. The main hurdles right now are cost, speed. But the cost of DNA sequencing has been plummeting for years, even outpacing Moore's law for computer chips. Engineers are working tirelessly to innovate, developing new chemical processes, building microfluidic lab-on-a-chip devices that will make writing and reading DNA data thousands of times faster and cheaper. A hospital could store a patient's entire lifetime of medical records. High-resolution MRI scans genomic data in a tiny, stable DNA archive that lasts forever. Our personal devices might have a biological coprocessor for long-term memory, backing up photos and documents into a format that will outlive us by centuries. Imagine a colony of bacteria that stores data and performs computations. We could engineer living hard drives to detect environmental changes and record them into their own DNA. To young aspiring engineers, you live in the most exciting time. The tools at your disposal: AI-driven design, CRISPR gene editing. The future is not just something you will live in, it is something you will create. Don't forget to like, share, and subscribe. ET Muster.